Okay, you're live.
Hello, everyone. Thank you very much for joining us today. We're happy to welcome you to the Human Cell Atlas Biological Networks seminar. Today, we're featuring the eye and pancreas bionetworks. This webinar is generously supported by Chan Zuckerberg Initiative. This is our agenda for today. We're going to start by a 15-minute talk featuring eye biological network, represented today by Joshua Sains. This will be followed by a 5-minute question and answer session. Please use the question the question function of the Zoom to ask your questions, and please upvote or downvote the questions that you see there. Then it will be followed by a 15-minute talk by Klaus Kessner, who represents Pancreas Bionetwork today, with 5 minutes of questions and answers with him. And then we go into 2 breakout sessions. We will post the links in the chat window. The eye bionetwork breakout is coordinated by Ru Chen and Ayala Segre today. And the pancreas bionetwork is moderated by Marcela Brisova and Roland Ailes. So with this, I'm happy to introduce our speakers. The first speaker is Joshua Sains from Harvard University. I hope you can see Josh.
Here I am.
Great. So just 1 second. So Dr. Joshua Sains and his colleagues study the structure, function, molecular architecture, and development of the mammalian visual system. To further their aims, they have also pioneered new ways to mark and manipulate neurons and the synapses they form. Joshua's current work focuses on comprehensive classification and characterization of neurons in the vertebrate retina using single-cell transcriptomics and the use of this data to address issues in development, injury, disease, and evolution. Joshua Sains received his bachelor's degree from Yale and PhD from Harvard, and then completed a postdoctoral fellowship at UCSF before joining the faculty of Washington University. He returned to Harvard in 2004 as professor of molecular and cellular biology and founding director of the Center for Brain Science. He is a member of the National Academy of Sciences and the American Academy of Arts and Sciences. He has served on editorial boards of scientific journals, including Cell and Neuron, on planning committees for the NIH Brain Initiative and the NEI Audacious Goals Initiative, and on advisory boards of the Max Planck Institute, Wellcome Trust, Stowers Institute, Hebrew University, and Howard Hughes Medical Institute. Joshua, welcome.
Thank you. Can you see what you want to see?
Please go into presenter. Yes, we do now. Thank you.
Okay. Well, great. Thank you very much. It's a pleasure to be here. So as part of the eye team, our job is to generate a cell atlas of the whole eye. But in fact, most of my lab's work has been on the retina. And I'm going to stick to that today, and in particular, focus on some very new studies on the evolution of retinal cell types. And by new, what I mean is woefully incomplete. But I hope that I can give you a sense of where we're going, and maybe more in the breakout session, elicit some suggestions for how we should proceed. So for those of you mired in the viscera, let me give you a little bit of an introduction to the retina. The main point here being that it is a part of the brain. And in fact, it's about as complicated as other parts of the brain. It's just been pulled out from the skull. And partly because it's been pulled out from the skull, it has a lot of advantages that enable more detailed study than other parts of the brain that might be more interesting intrinsically, for example, the cerebral cortex. Here's how it works. Photoreceptors in an outer cellular layer receive light and transmit information about illumination to 3 groups of interneurons in a middle cellular layer called horizontal bipolar and amacrine cells. They process the information and then pass it to the output cells of the retina, the retinal ganglion cells, that send their axons through the optic nerve to the rest of the brain. The key functional feature is that the photoreceptors are light detectors, whereas the retinal ganglion cells are feature detectors. They're not particularly interested in just diffuse illumination, but rather sensitive to particular visual features, for example, edges or color contrasts or motion. And the way they get their feature detecting ability is that each of the cell classes is divided into multiple types. And as a consequence, the information from photoreceptors is processed in complicated ways and fed in very specific synaptic circuits in an inner synaptic layer to the retinal ganglion cells, thereby endowing each of the types of ganglion cells with particular feature sensitivity. Now, just before we started the work on the cell atlas, the estimate was that the heterogeneity was to the tune of 65 retinal types. We went about isolating each of the three most heterogeneous classes so that we could get a comprehensive atlas. And that work's published. I'm not going to talk about it, but just make the point that at least in the retina, and to the extent that we've checked, the classification of cell types by molecular criteria is the same as the types correspond to those that are defined by criteria that neurobiologists would care about, which are physiological and morphological ones. And this slide just shows the results from bipolar cells, where we did the most complete characterization and comparison, where the molecular types correspond one-to-one to morphological types, as we found, and others have shown that light microscopy corresponds to EM reconstruction and to types from physiological high throughput recordings. So the upshot is that by the time we were done, the number of cell types in the retina that were known had doubled to around 130. There's another 10 to 15 non-neuronal types, so more than 140, approaching 150, which supports the contention that I made at the beginning, which is that the retina is indeed about as complicated as other parts of the brain. These numbers are similar to what one finds in a cortical area in the spinal cord and so forth. Now, there are lots of reasons that the number increased, but I think the doubling in most part is due to the advent of single cell sequencing and the sort of molecular cell atlas generation that the whole cell atlas program is designed to foster. So since we got this atlas, we've been trying to use it as a foundation to ask a series of biologically interesting questions about development, disease and injury, and what I want to talk about today, evolution. So the rationale for looking at evolution is that we know a huge amount about the conservation and differences of genes among species, but there ought to be a similar science to be devised for cell types. Which cell types are conserved, which ones change, which differ, how do they differ, and so forth. And we think that the retina is a good place to launch an investigation of that sort because its structure and its cell classes are conserved amongst essentially all vertebrates. There's a question mark that I put in after all, and that's not because I know of any exceptions, it's just that there may be exceptions that I don't know of. And that's quite different from brain areas, for example, the cortex, where it's hard to look at the evolution of cell types because the entire structure of the cortex differs fundamentally among species. And in fact, in some lower vertebrates, there isn't anything that you'd really call a cerebral cortex. So what I talked about so far is mouse. Several years ago, Irong Peng, a postdoctoral fellow in the lab, decided to look at primate retina. First, of course, we're primates, but particularly, although the mouse retina is a wonderful model for looking at the development and structure and function of neural circuits, it's actually a terrible model for looking at vision. And the reason is that most of our useful vision is mediated by a small central region called the fovea, only 1% or so of the retinal surface, embedded in a slightly larger region called the macula. Essentially, all of our high acuity vision comes from the fovea, and most of our color vision in daylight circumstances. And it's clinically important because blinding diseases, many of them selectively affect the macula, for example, age-related macular degeneration, which is the leading cause of irreversible blindness in the United States. And the point here is that primates are the only mammals with a fovea. And I think somebody can correct me that the fovea may be the only primate-specific structure in the whole mammalian brain. So at any rate, Irong separated the fovea from the rest of the retina, profiled both of them, and came up with cell atlases for the fovea and periphery, first for macaques, later humans, and she's now working on marmoset. And although I don't have time to go into it, that gave us a way to look at why it is or how it comes to be that the fovea is different from the periphery in structure, function, and connectivity, which had been documented by many groups over the years. And the upshot is that the cell types are largely shared. So the fovea isn't a fundamentally different structure. There's some proportions, but the biggest differences are in differentially expressed genes between shared types. And one imagines, and it's now testable, that among these genes are those that are responsible for the structural and functional differences that people have found. So that gave us the beginning of what could be an evolutionary comparison. And since then, we've moved on to profile retinas from a variety of other mammals, as well as chickens and zebrafish, and we're just getting going on lizards. And this is just a current census of the number of types that we found in these various species. So I'll finish with just two results. As I said, this is a very new study. One has to do with cell classes. So as I already said, the classes of neurons and the sole retinal glial cell, the Mueller glia, are perfectly conserved amongst vertebrates as far as we know, as is their function. But we didn't know whether that extended to the molecular level. And so here's an example that Wenjun Yan, a postdoc in the lab, generated, in which she reclustered 4 species, humans and macaques, 2 primates, mice and chickens, and then looked at the conservation. And what you can see in the dendrogram on the right, which shows transcriptomic similarity, is that indeed the cones of all 4 species are each other's nearest neighbors. They don't cluster by species, they cluster by class. Rods are the closest relatives of cones, and so forth down the line. Now for types, things are a little bit more complicated, a lot more complicated. And here again is Wenjun's analysis of these 4 species, where she now reclustered the amacrine cells alone and the ganglion cells alone. And if you look at the left, you can just see by eye that most of those clusters contain cells from the 4 different species, which are shown in 4 different colors. And the number of species contributing to each cluster is shown in the middle, more than 2 for amacrines, even more for bipolars. For ganglion cells, though, it's a different matter. You can see by eye that most of the clusters are only 1 color, and indeed, most of them receive large numbers of cells from only a single species. And so that suggests a hypothesis in which it may be that the outer retina, the photoreceptors and the interneurons, are a beautifully constructed graphics card that evolution has decided to conserve. And where evolution works to suit each retina to the needs of its species is at the level where information is passed from the eye to the rest of the brain, that is the retinal ganglion cells. And I'll finish with one more result that sort of supports that idea. And this is by Josh Hahn, a graduate student in Karthik Shekhar's lab, who now has Karthik now has his own laboratory at UC Berkeley. So on the left, you see the species I've been talking about in a conventional dendrogram of species similarity, evolutionary similarity. Here on the top, Josh has reclustered the bipolar cells from all of these species. And what you see is that their transcriptomic relationships to each other correspond pretty perfectly to the evolutionary relationships that one would expect. The primates are each other's closest relatives, chicks and zebrafish are outgroups and so forth. But it's fundamentally different for retinal ganglion cells. In fact, they're crazy. Zebrafish are more closely related to the primates than they are to, for example, chicks. Chicks are more closely related to rodents than pigs are related to rodents. Now, we don't really know what this means, but one intriguing idea is that maybe transcriptomic relationships come in large part from neutral drift. So the relationships of species by the transcriptome are essentially a clock that's giving us an estimate to how long it's been since a species diverged. Whereas for retinal ganglion cells, maybe what we're seeing are signs of positive and negative selection that override the relationships coming from neutral drift. And we don't know if this is true, obviously, and we don't know what the relationships mean. We have no idea why zebrafish come out closer to macaques than they do to other species or than mice do to macaques. But now we're in a position to first test this idea by looking at more species, and second, dig into what are the genes that are driving this clustering to see if we can gain some insight into how and why it is that the retinal ganglion cells seem to be evolved in a different way than other parts of the retina, presumably so that the needs of each retina is matched to the ecological needs of the species that hosts it. So I'll finish there. This has been a huge collaborative project. I don't have time to name everybody, but I'm just naming Josh Han, Karthik Shekhar, and Wenjun Yan, who did the computational work on the evolutionary studies that I described. Thank you very much.
Thank you so much, Joshua. Wonderful talk. Please do submit your questions via the question function in Zoom. We will wait a minute to see questions appearing. Nothing yet.
Well, are we panelists allowed to pose questions?
Absolutely, absolutely, by all means.
Because I don't think I can type into the F&A.
The panelists can ask questions as well. And if you're a panelist, please put it in our chat window and I'll copy it into questions and maybe someone will upload it. Go ahead.
Well, probably for the sake of time, I just make it verbally. So great talk, Josh. I have one question. So it was kind of amazing to see how the species cluster differently with respect to the different transcriptomic profiles of different cell types. So have you also attempted to take all the transcriptomic information from all the different cell types and redo the clustering of the species to see whether you get some more similarities with the regular expected species clustering?
No, Josh Han is doing that, but we don't really have the results yet. It turns out that, the annotation of the genomes varies among species. So there's a lot of stuff under the hood that Josh has to do before he can do a really good comparison of that sort. But yes, that's definitely ongoing and important.
I think there's also a question from Marcella. Go ahead.
That was a wonderful talk. I really enjoyed it because in pancreas field, there's controversy, mouse versus human. How much can we learn from mouse islets and translate that to human islet biology? So that was quite fascinating to see how we were traversing across different species. I was going to ask, so at transcriptional level, there's a lot of differences across species. Have you had a chance, or are you planning to also look at chromatin architecture across different species?
Yeah, that is a great idea. We would like to do that. I'd have to say it's, so people are doing a TAC-seq piecemeal on retinas from various species. And so I think some of that will come out. It is, yes, something we should do, but it isn't underway yet. I think they'll be probably pretty good at TAC-seq atlases from at least mouse and humans in the very near future.
Thank you.
Great. Thank you very much. If there are no other questions, I think we can go ahead and proceed with the rest of our seminar today. I am happy to introduce Dr. Klaus Kessner, who is representing the Pancreas Network today. Dr. Klaus Kessner is the Sir Butterworth Professor of Genetics at the University of Pennsylvania, Perelman School of Medicine, and Associate Director of Penn's Diabetes Research Center. Dr. Kaestner received his PhD from the Johns Hopkins University School of Medicine and completed a research fellowship at the German Cancer Research Center in Heidelberg. Dr. Kaestner's work is focused on the understanding of the molecular mechanisms of beta cell mass expansion and glucose homeostasis. In addition, his laboratory has pioneered the development of functional genomics tools applicable to the study of the endocrine pancreas. Dr. Kaestner is also founding member of the NIDDK-sponsored Human Islet Research Network Consortium, a group of 30 investigators whose common goal is to understand the molecular physiology of the human beta cell in order to allow for improved cell replacement therapy of type 1 diabetes. Dr. Kaestner has served on multiple review panels for the Juvenile Diabetes Research Foundation and for the National Institute for Diabetes, Digestive and Kidney Diseases. Dr. Kaestner serves on the editorial boards of the Diabetes Endocrinology and Molecular Endocrinology and was associate editor of the Journal of Clinical Investigation. Klaus, please go ahead. Welcome and thank you for joining us today.
Thank you so very, very much for the invitation to speak to you today. I'm speaking on behalf of a very large group of investigators who come together in the Human Pancreas Analysis Program, in short HPAP, and I'll tell you some of our work and a very high-level overview of how we go about to doing multimodal analysis of the human pancreas in health and disease. So what's the rationale for having the Human Pancreas Analysis Program? And it was decided by NIDDK in 2015 that the current models for diabetes are inadequate. Most of these models are rodent models and they have been very informative, but they don't model everything that happens in human disease precisely. And they also felt that because of these knowledge gaps we have in our understanding of the human pancreas, that actually makes it difficult to develop new methodologies to delay the onset and prevent type 1 diabetes and type 2 diabetes. So that's why HPAP was founded in 2016. So this is a mission statement for HPAP. HPAP is performing deep phenotyping of the human pancreas and its interactions with the immune system to better understand the cellular molecular events that precede and lead to type 1 and type 2 diabetes. The primary goal of HPAP is to accumulate, analyze, and distribute high-value data sets to the diabetes research community. There's a big distinction to another very famous consortium called the NPOD Consortium. It's shown at the bottom here, Network for Pancreatic Organ Donors with Diabetes, whose goal is to obtain tissues for distribution to researchers worldwide to do their own assays. HPAP is distributing data sets to the research community. And of course, all these are made available before publication. Shown here are some of the key members leading our effort. Of course, each of these has multiple people in their labs, so I really cannot put all the people on the slide here. Of course, Marcella, who's on our call, is one of the leaders in this program, together with Ali Naji, Al Powers, and Mark Atkinson. And here you see the institutions that are involved here. That's the University of Florida, University of Alberta, Penn, Vanderbilt University, and Stanford University. So really a large group of people trying to come together to further understanding of the human pancreas. So what do we do? This slide explains our workflow. Beginning of this is always the identification of human deceased organ donors that might be useful for our studies. And this is done jointly by investigators from HPAP, particularly Al Powers, who identifies organ donors in the type 2 diabetes field, and NPART, led by Mark Atkinson, who tries to identify organ donors who either have type 1 diabetes or are predisposed to getting type 1 diabetes by the fact that they have autoantibodies against beta cell proteins in their bloodstream. Once the screening is complete and identified organ donors, the organs are shipped to one central facility for processing. So both the pancreas and also the associated immune tissues, such as the pancreatic lymph nodes, the spleen, and so forth, are collected. And we have a very detailed protocol of what we do with the pancreas. So sections are obtained from the tail, the body, and the head of the organ for multiple modalities for tissue processing. And importantly, because the pancreas, the function we care about is that of the endocrine pancreas, the hormone-producing part of the organ, which is located in these islets of Langerhans. We also isolate pancreatic islets from all organ donors, so we can actually study the function, the metabolism, immunology, and of course, molecular profiles. So summarize them, we obtain both tissues as well as islets and cell suspensions. These most of the assays then are performed at Vanderbilt University and at Penn. This includes analysis of the tissue by various profiling methodologies, molecular profiling, looking not just at single-cell RNA-seq, single-cell attack-seq, but many others, islet physiology, multiplex imaging, and immune studies. So these are all done on the same organ donors, and these data are then collated into a database called TankDB, which collects all the data produced by HPAP. To give you a bit more granularity, show you this. Beginning, of course, we collect the donors, organ donors redacted medical charts, so a lot of information about the organ donor is available. And we, as I said, proceed with isolation of pancreatic tissue, the islets, and DNA. DNA is used, of course, for genotyping of the individuals. In fact, we perform whole genome sequencing, so not only mapping of genetic risk score, but even identification of new mutations is possible. We proceed with metabolic and physiological phenotyping of the isolated islets, as well as cellular molecular phenotyping and tissue morphometry and analysis, and even some biobanking for future assays. So on the islet front, we do functional phenotyping, insulin, glucagon, somatostatin secretion, calcium imaging, respiration. We also analyze the composition of the pancreatic islets using flow cytoph and perform electrophysiology, in particular patch-seq by Patrick McDonald in Canada, to actually match electrophysiological function to gene expression of single cells. The cell molecular phenotyping is done after cell dispersion. We either FACS sort the cells in the major constituents, which are the insulin-producing beta cells, glucagon-producing alpha cells, and acinar cells, for doing bulk RNA-seq, ATAC-seq, and DNA methylome analysis. But we also perform single-cell ATAC-seq and single-cell RNA-seq on all these donors. And of course, for tissue morphometry, we're doing standard immunostaining, but also multiplexed imaging, for instance, via Codex, done by Marcela Brissova and her colleagues, and imaging mass cytometry, which allows us to perform neighborhood analysis, niche analysis, and so forth. So we can actually then construct the three-dimensional architecture of the islets itself, the innervation, vasculature, and so forth. And as I said, all these data are then collated into TankDB. Importantly, for the image analysis, we actually also port the data to Pancreas Atlas, which go in a little bit more detail later on. How have we done thus far? As of to date, we've collected organs from 109 organ donors. 23 of these had type 2 diabetes, 22% had type 1 diabetes, and the rest had no history of diabetes. However, this group is actually subdivided into those people which have autoantibodies against islet antigens in their blood. So these are people at high risk of converting to diabetes someday, and a very important class of organ donors. And that's the age distribution. Clearly, of course, type 1 diabetes is the disease of more of young individuals, and type 2 diabetes of much older. So we actually, of course, have to have control groups for both, and we actually have to expand our control groups significantly to make this a better comparison in the future. What have we been able to produce? I think we actually have already been able to make several important contributions to the field. And this is one paper published in Cell earlier this year from our immunology group, which actually challenged an idea published the year earlier that there are special immune cells which are positive for both the T cell receptor and the B cell receptor. So if you wish, confused immune cells, that they would have a major role in type 1 diabetes, and work from HPAP could refute and challenge this claim, which was, I think, really important for the field. So I mentioned we developed flow cytoph to allow for precise quantification of cells, and published that in Cell Metabolism. And this is now used routinely on every preparation we have to count precisely not just the major cell type like the alpha and beta cells, but also the minor cell type, endocrine cell type in the islet, as well as profiling the immune cells that might find themselves within the pancreatic islets themselves. We also developed spatial proteomics. So this is for doing imaging mass cytometry, which we published first for people with type 1 diabetes, and more recently also for type 2 diabetes. So clearly, using this technology to gain much more information on the human pancreas in health and disease. Several more papers are in the, this is important. I mentioned earlier that we've been very fortunate that we can port images to Pancreas Atlas, a platform developed by Dan Saunders, Marcela Brisova, and Al Powers. And what's so cool about this is that any user in the world can take an image and say, well, today I would like to look at 3 of these channels, and I define which colors I want to show and create a new image. They can change the intensity of each color and of each channel. So really analyze the imaging data in any way they choose, and literally in one day they could create millions of different overlays of these spatial proteomics data. So wonderful that this was possible. Some additional studies in the pipeline. We've have a paper coming out in Nature Metabolism on multi-omic single-cell analysis of the pancreatic islets. This combines single-cell RNA-seq, flow cytoph, and imaging mass cytometry in one study, and that should be quite interesting. Very recent work from Marcela, Doris Stoffers, and my group showed that in the very earliest stages of type 1 diabetes, when there's just autoantibodies, there's even already detectable alpha cell dysfunction, which is really another interesting clue about the pathogenesis of this disease. And finally, very recently, we put into bioRxiv a paper where we could combine single-cell transcriptomics, ataxic, and high-C analysis to actually determine the 3-dimensional chromatin structure, and thereby see that the genetic risk for type 2 diabetes is actually shared by multiple cell types within the pancreas, not just the beta cells, but alpha cells and Kaestner cells as well. So to summarize what I told you, HPAP established a nationwide infrastructure to procure the pancreas and immune system to understand the pathogenesis of type 1 and type 2 diabetes. We adopted and developed state-of-the-art assay modalities to maximize the information that can be gained from each of these rare organ donors. And we already discovered information that provides new insights into type 1 diabetes and type 2 diabetes pathogenesis. Finally, we've heard about TankDB. So that's the publicly accessible database where all our data are deposited. And as I said, we deposit data here way before publication. And so you might find this useful to access additional information. And finally, I want to thank you for letting me speak to you. And as I said, I'm speaking on behalf of a large group of investigators that all have made major contributions to our work. thank you. I'm happy to take questions.
Thank you so much, Klaus. Waiting for the questions to appear in the question function of the Zoom talk. Meanwhile, panelists, if you have any questions to Klaus, we'll be happy to take them.
Klaus, this is Roland. A great talk and a great overview about the different activities. I just wonder, since you have this fairly large control group of 55 individuals to match both type 1 and type 2 diabetes in a wide age range, if you just look at the normals, so to speak, I mean, how many abnormalities do you find in the normal controls? And secondly, how much does this kind of normality changes over lifetime?
Yeah, thanks for that point. It's very interesting. You're right that these data sets should in principle, or do in principle, actually enable this kind of analysis, because we even have control runners of very young age and then all the way to 65 or so. I should have to confess that as of today, nobody from the HPAP group has undertaken that particular analysis. But as I said, the data are available. What does happen, however, in parallel, Marcela Brisova and Al Powers lead a great effort in the HANDLE program, where they actually look specifically at the human aging, or the pancreas during human development between birth and I think 10 years of age or so. So there are a lot of data sets available of this type to actually study human ontogeny of the pancreas. Yeah, so it's a very good point. We have not done that. I'm not aware of anybody within our big group who's actually specifically focused on that or used the data for that purpose. That's a good idea, though.
Thanks, Klaus. We have a question from Chris Sander. Do you have any information about predisposition to pancreatitis or pancreatic cancers?
So that's an interesting point. What we have not done, for instance, is take the pancreas tissue and look for mutations in KRAS and the like to see if there's predisposition. On the other hand, we have discovered on occasion in several just looking at the H&E histology, precancerous lesions in either chronic pancreatitis or early Panin lesions. So there's a pathologist that looks at all of the H&E sections of these to actually document that. So there might be a chance actually to search the data in that fashion to pull out those which actually have early pancreatitis and early changes towards pancreatic cancer. Yeah, good point. Can be done.
Thank you. Any other questions to Klaus? Please post in the question function of Zoom. Okay, if there are no other questions, or if you think of your questions a little later, you'll have a chance to ask them during during the breakout session. So if you remember, we're going to go into breakout sessions. They are about 45 or 40 minutes long. And if you're not going into breakout sessions with us, thank you for joining today. You will be able to see the recording of the main session that we just had on our YouTube channel. Oh, there is one question. Let me see. I think Chris has raised his hand, and that might have been an older question. Okay, so with this, please proceed to your breakout room of choice, and thank you very much for joining us today.